Hello all. This is the video segment for Chapter 22. It's all about anaerobes of clinical importance. So let's talk about some anaerobes. So by definition, an anaerobe is a bacteria that's able to grow and replicate in the absence of oxygen. They vary in their response to the presence of oxygen, and there's three kind of varying degrees. They each have a title. The first one is an obligate or a strict anaerobe, and this organism type is killed almost immediately in the presence of oxygen. The second type is a moderate anaerobe, also known as aerotolerant. These can survive in the presence of oxygen, but cannot carry out important metabolic functions unless they're in an anaerobic environment. And then the third degree or group is a facultative anaerobe. And this type of an organism prefer to use oxygen, but can grow and carry out metabolic functions without oxygen. So let's talk about where anaerobes can be found. So there's two primary groups based on where we find anaerobes. The first one is exogenous anaerobes, and the second one is endogenous anaerobes. So exogenous anaerobes are those naturally found outside of the body of animals. Infections are usually caused by gram-positive spore-forming bacilli belonging to the genus Clostridium. And an infection is usually initiated when a spore-contaminated food is ingested or an open wound is contaminated with soil. And then that second group, the endogenous anaerobes, are anaerobes that exist naturally inside the body of animals. And infections with this type of an organism are usually the most common type of anaerobic human infection. And these are capable of causing disease in any anatomic site of the body. So that leads us to anatomic sites of infection that you can find an anaerobic organism growing in. So there are about four main areas of the body. The first one is the respiratory tract. 90% of bacteria in your saliva and nasal washings, gingival and tooth scrapings are anaerobes. Therefore, an expectorated sputum or an oral swab are unacceptable for anaerobic culture. Next site is skin, and your number one anaerobic organism found on the skin that usually causes a contamination in our cultures is Propionibacterium acnes. This is a skin colonizer. So the presence of Propionibacterium acnes in any culture usually represents contamination. The next location or anatomic site is the genitourinary tract. Anaerobic bacteria colonize the distal urethral areas. 50% of bacteria in cervical and vaginal secretions are anaerobic. And genitourinary tract swabs and voided urine from catheters, these specimens are unacceptable for anaerobic culture. And then that fourth group, that's the GI or gastrointestinal tract. Anaerobes definitely outnumber facultative anaerobes, about 1,000 to 1 in your GI tract. So stool is not routinely cultured for anaerobes. There is an exception, and that's with the isolation of Clostridium difficile. So there are some factors that can predispose a patient to an anaerobic infection. Some of these factors include trauma to the mucous membranes or skin, an introduction of soil to an open wound, or puncture of the GI tract in any way. This is some examples of trauma that can introduce anaerobes to an area they're not normally found. Also, locations with vascular stasis or poor circulation. So tissue sites with poor circulation usually don't have very much oxygen. So this is a good place that would predispose a patient to an infection if an anaerobe was able to set up shop there. Tooth extractions and tissue necrosis are all predisposing factors as well. So some indications, if you were to have an anaerobic infection, some indications would include a foul odor. Also, sometimes there'll be large quantities of gas in that infected tissue. Something called sulfur granules may be present, or they're like clumps of organisms. And then something called polymicrobial infection. And this is where the presence of one organism creates a niche for another pathogen to colonize and cause infection. And then infection persists. If an infection persists despite of an aminoglycoside antibiotic wound therapy, this possibly this kind of can be an indication that there's anaerobic involvement. So there are 4 groups of anaerobes based on gram stain and morphology. So if we break them into those groups based on those 2 things, we have gram-positive anaerobic bacilli, we have gram-negative anaerobic bacilli, and then we have gram-positive and then gram-negative cocci, anaerobic cocci. So the first group that we're going to talk about each one of those 4, but the first group is the gram-positive anaerobic bacilli. And in this group, we could further split it, branch it into 2 smaller groups, and that would be spore-forming and non-spore forming. So of the spore-forming gram-positive anaerobic bacilli, most of these are Clostridium genus. So let's talk about those first. So our gram-positive spore-forming anaerobic bacilli all are classified in the genus Clostridium. Clostridium may be grouped according to endospore location. Remember, we have terminal, central, subterminal location. There are 5 main organisms that we're going to talk about. The first one is Clostridium perfringens that causes food poisoning. There's another Clostridium perfringens, so it's kind of 2 types of this organism. That second type of perfringens causes myonecrosis or gas gangrene. So we're going to talk about each one of those. Even though they're the same kind of organism, they present very differently. We have the next group or species, which is Clostridium botulinum, and then we have Clostridium tetani and Clostridium difficile. So let's talk about Clostridium perfringens, the food poisoning type first. So before I go any further, I want to mention 2 types of tests or agar. And the first one is the egg yolk agar. This is an enriched differential medium that is very useful for differentiating Clostridium species. And how it differentiates is based on lethisinase, lipase, and proteolytic activity. So, for example, Clostridium perfringens, the food poisoning here, The alpha toxin that it produces performs phospholipase activity on lecithin, and this creates a zone of opacity surrounding the growth. So this particular organism has a large zone of opacity on the egg yolk agar. There's another test for lithicinase, and this is called the direct Nagler test. So this one is another test for lithicinase. Okay, so let's talk about Clostridium perfringens. It's associated with two types of GI illness. The first one's type A. It's a mild presentation, self-limiting GI tract illness. And then there's a type C, and this is more serious. This is a rarely seen GI tract illness. So usually this disease follows ingestion of an enterotoxin producing strain in contaminated food. And without treatment, type C illness is often fatal. And even with treatment, type C illness can be fatal as well. I think even with treatment, it's like 15 to 25% mortality rate. Clostridium perfringens is a large gram-positive boxcar rod. It does not swarm on blood agar plate. It has this unique characteristic double zone of hemolysis on an anaerobic blood agar plate. And when I say a double zone of hemolysis, it's talking about there's a primary first zone directly around your growth, and this zone has complete hemolysis in it. And then there's a second zone on the outside of that first zone that has incomplete hemolysis, and that's caused by the alpha toxin that this one produces. So that's Clostridium perfringens food poisoning. Let's talk about Clostridium perfringens myonecrosis or gas gangrene presentation. There are actually several Clostridium species that can cause myonecrosis, but Clostridium perfringens is the most common cause. Well, usually this occurs with organisms that contaminate wounds or either a trauma or a surgery. They enter through trauma or surgery. This organism produces an exotoxin that causes the necrosis and allows for deeper penetration of the organism. Affected areas often require amputation with this organism. So that's all I want to say about perfringens and the myonecrosis. Just know that there are two forms of disease that perfringens causes. That's food poisoning with the two types of GI illness, A and C, and then gas gangrene and the myonecrosis. So let's move to the next species of Clostridium, and that's Clostridium botulinum. This particular organism causes botulism. This is a food-borne illness that results from the ingestion of preformed botulinum toxin. It's an extremely potent toxin. A very small amount can cause paralysis and death. Common food sources include home or canned vegetables, cured meats, fermented fish, and other preserved foods can also harbor this toxin. There's something I want to mention called infant botulism, and this involves the ingestion of spores. Most common source is contaminated honey. So if you've ever heard a pediatrician tell you or your child or someone else to not eat honey before especially the age of 2, this is why. Infant botulism. On an egg yolk agar, this particular species is lipase positive. It also has a gasoline or mother of pearl appearance. So that's kind of a unique trait. So that's Clostridium botulinum. The next organism is Clostridium tetani. This causes tetanus. This particular organism produces a very strong neurotoxin called tetanospasmin. This tetanospasmin causes continuous muscle spasms, or also known as lockjaw, and also causes breathing difficulties. It's not very common in the U.S. due to widespread DPT vaccine, which is diphtheria, pertussis, and tetanus vaccine. So thankfully, we've had that vaccine to kind of limit the number of cases we have in the U.S. There is another disease I want to mention caused by this organism called tetanus neonatorum, and this is tetanus in a newborn baby. It's possibly due to contaminated instruments during the birthing process, or umbilical infections, or sometimes seen in cases of septic abortions. These are gram-positive rods with very square ends. Some are gram variable. These swarm on an anaerobic blood agar plate, and they have terminal spores. Some of those little characteristics about Clostridium tetani. And the last Clostridium species is Clostridium difficile, and you will definitely see this quite often in the laboratory, the clinical laboratory. Let's see. This is called the antibiotic-associated diarrhea, and pseudomembranous colitis is oftentimes the result of this organism overgrowth. We are normally colonized with this organism in our GI tract, but when you have long-term antibiotic treatment, or a patient has long-term antibiotic treatment or a heavy dose of antibiotics, it can wipe out your good flora in the intestine, and this particular organism is an opportunistic pathogen, so it can take over and cause some serious problems. It produces two types of toxin. It produces toxin A, which is an enterotoxin, and it produces a toxin B, which is an exotoxin. You'll often see bloody diarrhea associated with necrosis of the colon mucosa, and it is a common cause of nosocomial diarrhea. There is an agar called cycloserine cefoxitin fructose agar, which is abbreviated CCFA, and this is selective and differential for C. diff. These organisms will fluoresce chartreuse under UV light, and they sometimes have an odor of horse dung, which is kind of unique to this organism. So those are your 5 Clostridium species, and that wraps up our gram-positive anaerobic bacilli for the spore formers. So let's talk about the gram-positive non-spore forming anaerobic bacilli. There are 6, I think, mentioned in your PowerPoints, 6 groups that we'll briefly discuss. The first one is actinomyces. Actinomyces causes the disease actinomycosis. This is a chronic granulomatous infectious disease. Let's see. There are a couple species that are more prevalently seen in the clinical lab. One is Actinomyces israelii. So this particular disease caused by Actinomyces species are characterized by the development of sinus tracts and fistulae, which erupt to the surface and drain pus that contain sulfur granules. These actinomyces species can cause infections to the brain, to orofacial region. They can cause pleuropulmonary region infections and can even infect the genital organs. And a term sometimes associated with the orofacial infections caused by actinomycosis or actinomyces is lumpy jaw. So that's a kind of a strange term, but it's called lumpy jaw. They are, or organisms in this group are slightly curved gram-positive rods, typically seen as branching. They stain irregularly, and because they stain irregularly, they tend to have this beaded or banded appearance on a gram stain. Young colonies have been noted to appear like a molar tooth, or they have this woolly or spider-like appearance on agar. So know those little characteristics of actinomyces. The next little group of non-spore formers is Mobiluncus species. Mobiluncus species is one of the organisms associated with bacterial vaginosis. There's usually or probably a synergistic infectious process that involves a variety of organisms that causes vaginosis, but most commonly it's associated with Gardnerella vaginalis species and Mobiluncus species, one of them. You can, when you look, at a wet prep with saline under the microscope, you will see that classic clue cell with a bacterial vaginosis disease state. And clinical features include a grayish-white foul-smelling discharge and little or no discomfort and inflammation. The next group of non-spore formers, Lactobacillus. These are highly pleomorphic bacilli, very important to the genitourinary tract in females. They're a good and important flora. These species lower the vaginal pH and suppress overgrowth of some of the bacteria that can be harmful if it overgrows, or cause infection if it overgrows. So infection by this group of organisms is highly unlikely. They're noted as good bacteria. The next group is Propionibacterium species. And these are usually seen as a contaminant in blood cultures. They're normal flora of the skin. They're anaerobic, of course, and they appear as dipteroid rods, more so like haystacks than the Chinese letters. They're catalase positive and indole positive, so a couple things to help identify these. And there's a couple others in this non-spore forming group that I'm just going to mention, but we're not going to talk about. And that's Bifidobacterium, which is a normal flora, or is part of the normal flora, and then Eubacterium. So just be familiar with those groups as well. So let's move on to anaerobic gram-negative bacilli. So let's move on to the gram-negative anaerobes. There are 4 species groups. The first one is Bacteroides. Second one is Porphyromonas. Third one is Prevotella, and then the fourth one is Fusobacteria. So let's talk about Bacteroides. One important organism I'm going to mention is Bacteroides fragilis, and this is often associated with soft tissue infections. It's a bile-tolerant group, and this organism species, Fragilis, is the most common species of anaerobe bacteria isolated from infectious processes. It's capable of producing beta-lactamase, so it should be tested. Isolates should be tested. And they produce brownish black discoloration around colonies on a BBE agar or Bacteroides bioesculin agar. And also in a esculin hydrolysis test, it will turn the medium from yellow to brownish black. There is a agar called KV Laked blood agar, and that includes kanamycin and vancomycin. And this is a choice agar for isolation of anaerobic gram-negative rods, especially Bacteroides and another group we'll talk about, which is Prevotella. So know that that is an important agar for these gram-negative rods that are anaerobic. The next group is the Porphyromonas. These species, there's two I'm going to mention. That's Porphyromonas gingivalis and Porphyromonas endodontalis. And these are associated primarily with oral infections. They're biosensitive as well. They're a pigmented species. And they can also produce brownish black colonies on BBE, and sometimes they fluoresce. The next group, Prevotella. There's one organism in this one I want to mention, and that's Prevotella asaccharolytica. This is biosensitive pigmented species, widely distributed in humid tissues and fluids. Certain species of Prevotella produce something called protoporphyrin, and this causes colonies to turn brownish black with age. Non-pigmented colonies in this group may fluoresce brick red under UV long wave light. And then the last group of species that I'm going to mention that are anaerobic gram-negative bacilli are the Fusobacterium. These are gram-negative, fusiform, long, thin rods, slightly pointed on the ends. Most of the time they're found in mixed oral infections. Some species, well, there's one particular species that I want to mention, and that is Fusobacterium nucleatum. And this is the only organism that is consistently fusiform. Most of the others are pleomorphic, long, thin gram-negative rods, but this particular species, nucleatum, is consistently fusiform shape. So that is the gram-negative anaerobes that are bacilli. So let's quickly talk about anaerobic cocci. So we have gram-positive cocci that are anaerobic and gram-negative. So the first group, gram-positive, there are three main groups I'm going to mention. One is Peptococcus species. Second one is Peptostreptococcus, which is usually a contaminant. And then the third one is Gamella species. But really, the Peptostreptococcus species are the most, are most of the time the culprit that we see in the lab. Two Peptostreptococcus species that I want to discuss and just give you a couple details about are Peptostreptococcus anaerobius and Peptoniphelus asaccharolyticus. So the former, Peptostreptococcus anaerobius, is presumptively ID based on gram stain. It's gram-positive cocci, but it also is SPS disc sensitive. So the sodium polyenethyl sulfonate test will inhibit growth of this organism. So it is sensitive to this disc. And then the Peptoniphelus asaccharolyticus is presumptively ID'd based on gram stain, gram-positive cocci. But however, this is SPS disc resistant. And it is indole positive. So Those are the couple details I wanted to mention about those. Moving on to the last group, the gram-negative cocci that are anaerobic. There's one clinically important species genre, and that's Veillonella. This is really our only pathogen to humans that is gram-negative cocci that's an anaerobe. There are several genera of anaerobic gram-negative cocci, but this one is really the only species group that is pathogenic. We presumptively ID it with the gram stain, gram-negative cocci, and it is nitrate positive. When it comes to antimicrobial susceptibility and testing on anaerobes, it's quite costly and time-consuming and labor-intensive. So there are kind of some requirements in order to do testing on anaerobes. Only agar dilutions and broth microdilutions are approved methods by the Clinical Laboratory Standards Institute. And there are only certain infection or infectious processes that warrant susceptibility testing. And these can include bacteremias, brain abscesses, tissue cultures, infections with prosthetic devices, endocarditis, and osteomyelitis. But I want you to note that all Bacteroides and Prevotella species, or isolates, are tested for beta-lactamase. So just know that. Bacteroides and Prevotella tested for beta-lactamase. And that is Chapter 22, Anaerobes of Clinical Importance.